I'm going to present some results about the project which we have done in the past 2 years on the cover crop strategies. So our focus is going to use cover crops to improve soil health, increase the sustainability of orchards, and also like improve the nitrogen management in the orchards. So today's talk, I'm going to focus on nitrogen. So first let me introduce my research team here. This is kind of multi-organization collaboration project. We have people from UC Davis, especially like Diana. She was my postdoc and she contributed a lot to this project. And also we have people from UCNR, like Jenny Hesse and Catherine, France, and Mo. They helped me dramatically at the beginning of the project and also during the implementation of the cover crops during the project. And also we have collaborators from the USDA ARS, like Carrie Steenworth. And also I want to specially thank the grower collaborators, Dominic and Roger, their generous contribution to this project to make this project being completed successfully. All right, so in today's talk, first I will introduce give some background on the nitrogen cycle in orchard. And also I will present the goal of this project. After that, I will give an example of the field trial which we have in this project and present some data, like preliminary data which we got out from the field trials. All right, so nitrogen cycling on an orchard. So from this graph, you can see in the orchard, so we put the fertilizer into the soil. Actually, fertilizers, the nitrogen get into soil. There are a few sources. One is through the synthetic fertilizer, and also we could go through like cover crops, like legume. They can do the plant like some nitrogen fixation legumes, so that will input the nitrogen for the nitrogen from the air and get into the soil. And also we have atmospheric nitrogen deposition into the soil. So after this part of the nitrogen get into the soil, and they will part of them can be used by trees and to produce yield and to get into different parts of the trees. But also this part of nitrogen will go through the different processes. For example, like immobilization, when the soil carbon is very high, or you apply with, for example, like Luke introduced, if they have orchard recycling, you have a lot of carbon input there. So part of the nitrogen will be incorporated into the soil, become like unavailable temporary. So that is the immobilization process. And if you put ammonium, for example, if you put urea or UAN, and that will go through the nitrification and become nitrate. Nitrate is also part can be used by the trees, but also nitrate will be subjected to leaching. So this is another concern from the water groundwater aspect, the water quality. And also the nitrate, when they have, like, for example, they have flood irrigation, the soil condition become very anaerobic. They can go through the denitrification and then produce some gas and get into the air, which will result cause other kind of concerns like global warming or like green the air quality. For example, NOx, if you produce that can cause some respiration issue. Okay, so in Walnut orchard, currently we have the nitrogen. I checked that from this CDFA, like their fertilizer recommendation website. The nitrogen application recommendation is kind of vary between about like 75 to 150 pounds per acre, depends on the soil, depends on the source of nitrogen, and because like our collaborator, like Catherine, and she published the paper like a few years ago and found that they have only when we like harvest walnuts, actually we only about they kind of remove about 50 tons per, like up to 50 tons nitrogen per acre out of by the harvested yield. So that means like every year we put 150 pounds per acre nitrogen into the orchard, but actually we only remove about 50 tons per acre. So this is kind of like we have relatively low nitrogen fertilizer using efficiency in all the orchards. So to understand, so how this is really a question for us as a researcher, like how can we improve this part of nitrogen use efficiency. To understand that, we will need to look at like nitrogen distribution in orchard, like where did nitrogen go and where it will go, where it is stored. And also it's like combined with the current like soil health management practices, especially cover crops. And we try to understand the nitrogen management in the whole orchard using cover crops. Okay. So the goal of this presentation today is like first we are going to quantify the nitrogen contribution of the different cover crops in walnut orchards, because in as some of you might know, like cover crops, they have not just one species, they have so many different mixes. Depends on your purpose and depends on the condition of your orchards, depends on what's your goal, and they have so many choices of the cover crops. So we trying to understand the nitrogen contribution of those kind of different mixes of cover crops. And also we want to understand the distribution of nitrogen in soil using cover crops. We want to say it's like how much nitrogen come out, like we're stored in the soil, how much nitrogen will get out of the system, and how much soil will be, for example, has potential leach to the soil profile. So here I'm going to give you an example of our field trials. In this project, actually we have 3 field trials. We have one in the coast soil lab, and we have one in the river garden farms, and also we have one down to the south in like nearby Lodi. And here we present the one results from, our results from this one trial is River Garden Farms, which is located like nearby the Leolo and the Colosa County. And in this orchard, the trees were planted in 2015. So this orchard is relatively young. The nitrogen management in like nitrogen fertilizer they put in 2020 is about 140 pounds per acre. And they have 2 applications. One they do the sprayer, and they spray their fertilizer to the trunk. So only like 3 lines received the nitrogen. And the second one, they did the fertigation, which they have the micro sprinkler, and so it's like covered the whole floor. And in 2021, we didn't put any nitrogen there. It's because the tree leaf analysis from 2020 showed that the nitrogen in the leaves is kind of above the optimal nitrogen level. So in 2021, we decided we're not going to put any nitrogen into the trial. All right. So we implemented 3 treatments. We tested 3 cover crop mixes. So one is legume, and the legume this consists of like different varieties of clover, and we also mixed grass and the legume, like 50% grass and 50% legume, and we also have the multi-species mix. The one of the multiple species mix, the multiplex mix is like this treatment was chosen by grower because they really want to see the biomass. They are very interested in the soil carbon returning to orchard, and this species is can produce a lot like a biomass. So we tested those three cover crop mixes. In 2020, we planted in November, right after the walnut harvest. But in 2021, actually the time was off. So this is 2019, we planted in November. And then in 2020, we planted in December. So it's relatively late. So first, I show the results is the biomass production. So for the biomass production, we have the 2 years. So from this graph, first we see the top one. So these brown bars represent weeds from each treatment. And for the residential, we call it control. It's just residential vegetation, which we didn't do anything. We didn't like apply the herbicide, or we didn't do like mow the field or doing anything. Just leave the row between the tractor row like grow by itself. So from this one, we can see the brown, this brown bar, like in the cover crop treatments, the weeds biomass is much lower. So in other words, if we plant cover crops, it can just help us to suppress, control the weeds. And also, if we look at the cover crop treatments, those kind of different corners are different species. As you recall, in the different, we have different like a species mix in the different cover crop treatments. So this is clover. This all biomass are from clover. And this one we have about like a little more grass than clover in the grass and clover treatment. And in this multiplex treatment, the most dominant species which grow very well was like brassica, and also they have veg grow pretty well, and peas next, and also they have oats, triticale. And in the second year, you can say the because second year we planted the cover crops relatively late, and also as you may recall, we experienced drought. We didn't get enough irrigation, even though we turned on the irrigation later in the season, but it still was not able to. The clover was not very, was not established very well. So you can see from this treatment, the width was not very significantly suppressed in the clover, and also like a grass treatment. But in multiplex, the width was significantly suppressed. And also the biomass is like relatively lower than what we got from the first year. So, and the highest biomass is still from the multiplex, which this is kind of match the expectation from the for these treatments. Okay. So based on the different species and their carbon nitrogen content for each species, we calculated how much carbon and how much nitrogen that will be put into the orchard by the time we terminate the cover crops. So here we can see the different the contribution of carbon and nitrogen from the different treatments. So here we can see it's like a multiplex because the biomass is highest in this treatment. So the carbon input is significantly higher than other treatments. And for the nitrogen input here, so we can say, yeah, it's because of they have high biomass, and also they have some certain part of the legume species there. So the nitrogen input is also like higher than other treatments there. And we haven't finished this data collection yet, but I used the reference which published in 2010 to talk about nitrogen fertilizer replacement value of cover crops. And in this paper, they talk about what's the nitrogen content from each species, and also what's the nitrogen fertilizer replacement value. So I did the calculation. It's come out about 45% to 50% of the total nitrogen can be, can contribute to the nitrogen fertilizer, can be replaced. So by applying that number, I did a very rough calculation here. So for the clover, and in a good year, and we can say we can have about maybe 45 pounds per acre nitrogen can come into the orchard. And also for the grass clover, we have 25 pounds per acre, and for multiplex, we about 70 pounds per acre. And even for the control, which we didn't do anything, but here I want to point out that in the control, because the site, actually we had the contamination on the control because they have a lot like burnt clover, which is like, transported from other site and get into that area. So in the control treatment, and they have a lot of burnt clover, so which we found some nitrogen in the control, which they make sense of there. So, but again, this number just a very rough calculation. This just gave us the demonstration is like relatively how much nitrogen we can get into the orchard. But another thing I would like to call your attention is also because this is nitrogen in the biomass, right? But when the biomass grow and they can pour the nitrogen into the plant, and for the legume, they fix nitrogen. That's true. But for the multiplex, they have some other kind of species. They just pull nitrogen from the soil. So those part of nitrogen, which we talk about returning into the orchard, actually part of them are in the orchard. Should not be considered as the excess external nitrogen there. And also another thing I would like to call your attention here is in 2020 and 2021, and we can say the clover, the nitrogen input in the clover, they're very stable. And you remember, it's like we have the nitrogen biomass. I mean, the clover biomass was not, even the clover was not established very well in the second year, but the nitrogen return actually was not reduced. But if you look at the multiplex, the first year was established very well, and they have a lot like nitrogen input, biomass input, but the second year it dropped. So in other words, those kind of varieties, those kind of mix, clover is more sustainable, like regardless of the climate. But the clover is kind of relatively more sensitive to the climate. If in a dry year, if you don't establish biomass very well, so the nitrogen content, I mean the nitrogen input or carbon input will reduce a little bit. Okay. So let's look at the nitrogen depth, nitrogen distribution in soil depth. So we collected the soils down to the about 90 centimeter, and we're trying to say is like, okay, we're trying to understand, okay, so this nitrogen on the surface or the nitrogen is distributed along the soil profile, right? So first we collected the soil from cover crop area, from the cover crop rule. This is by determination, either right before termination or the day right after. So it's like before the cover crop started to decompose. So this is results kind of very interesting. We saw the clover. So we did clover and the grasses treatment was not along the soil profile. They didn't change that much. The nitrogen distribution is kind of pretty uniform. For the multiplex and the control, they kind of have most of nitrogen distributed on the top 20 centimeter, but when you're going down, it's kind of get lower. And in the second year, it's kind of like, so the clover, because second year we didn't put any nitrogen there. So the clover and the grass treatment is kind of have higher. Actually, they all kind of have a little higher in the first top 20 centimeter, and then it's going down, down to the 90 centimeter. So it's kind of decreased. And at the harvest, by the time harvest, which the cover crop has been decomposed, about 4 months. Now it's like we look at the nitrogen in the soil, we can say it's the same. It's like we have more nitrogen on the surface, and they have less nitrogen down to the soil profile. And the treatment is clover and the grass. They have relatively higher nitrogen than the multiplex and the control. And in the second year, the trend was not very significant. And next, we collected also the tree line, because the cover crop is like really in the middle of the row. So you can see here, and there it's all like bare soil, like nearby the soil, like by the tree trunk there. So we collect the soil in tree line to analyze the nitrogen. So this trend is kind of in the opposite than in the then in the cover crop row. Cover crop row is all the nitrogen on the surface, and then going down is decreased. But in the tree row, is we have the lower nitrogen on the surface, and more nitrogen got into the deeper soil layer. So both the first year and the second year, and this is by the termination is in May. But if you look into the November, it's following the same trend. So this is kind of in tree line and the cover crop line, the nitrogen distribution along the soil profile are quite different. And so this is kind of very interesting phenomena to us. And our interpretation is like, we thought about maybe this is because of the, in the tree line, they don't have the, like vegetation on the surface. So all the like nitrogen starting to leach down. But in the cover crop is kind of help you to retain nitrogen on the top soil layers. So keep the soil over there instead of leaching down. So that's our like interpretation about this result here. Okay, we also looked at the Incising nitrogen organization, inorganic nitrogen in the top 30 centimeter. So this these different corners are different cover crops, and we have collected soil samples in a different date. So to see it's like how much of the nitrogen will be released from different cover crops. So from this graph, we can say after we terminate the cover crop and they have more like the legume, the clover treatment is kind of started gradually to increase their nitrogen availability in the soil. Nitrogen release like more nitrogen over the time. And also in the second year it's the same. So you have we have a lot like nitrogen from the clover mix and from the beginning, and this is kind of keep pretty stable over the growing season. But for the multiplex, and they have a lot of nitrogen released at the very beginning, and then when we get later and later, the less and less nitrogen is like being available. So for the cover crop, those mixes is like clover has a constant nitrogen supply, and also it's like we have like nitrogen availability being increased in the late growing season. And also inorganic nitrogen, we calculated it's like compared to the control, how much nitrogen is contributed by cover crops. And this is by using our data in the cover crop treatment, subtract the the difference, calculate difference between the cover crop treatment and the control. And from 2020, because we have fertilizer applied, so the difference is kind of not very significant, so it's kind of hard to tell. So let's look at the 2021, and we can see in the July, here in the July, we can see the most clover and grass supply their nitrogen most in July, and in July in both clover and grass, and after that in August, and then by the time we harvest, this is September, and by the time we harvest, we barely see any nitrogen being supplied. So the decomposition of nitrogen, the decomposition of the clover, the capability of the clover supply nitrogen is kind of match the growth season, walnut growing season. And the nitrogen leaching potential here, we looked at the nitrogen leaching potential during the year. We can see in the summer, the nitrogen leaching potential is pretty high. This we only looked at the cover crop area. That's because we have irrigation there. And in the fall, the nitrogen leaching potential is the lowest one, and also the grass treatment is very low, is lower than the control and the multiplex. And in the winter, when they have the when we have the rainy season, and we found that the statistically not very significant, but we did say there is a trend. We have more nitrogen can subject to leach in the multiplex and the control compared to clover and the grass. And the last one I would like to present very quickly on this one is 6-year orchard and 22-year orchard, because we have 3 orchards, and we found that it's like the nitrogen leaching potential is kind of relatively to, it kind of have something to do with the tree age. And in this young orchard, in cover crop area, and the nitrogen is kind of much lower. This is the soil profile, and the whole soil profile nitrogen content is much lower than the tree line. But in the older, 22 years old orchard, it's kind of opposite. So you have more nitrogen stored in cover crop area, but then tree line. So this is quite interesting to us. We are still trying to understand if this is really because of the tree age. Because in the young tree, and they maybe still establish their roots, and they don't take that much nitrogen. And so the nitrogen is being leached down to the soil profile, or if there is other reason, for example, maybe there is the irrigation or fertilization methods difference. So we're still trying to explain what's going on over there. Okay, the preliminary conclusion of this research is nitrogen inputs at cover crop termination. So we have the highest biomass from multiplex, and then we have clover, and we have grass clover mix, and we have the residential vegetation can also hold nitrogen. This part nitrogen is that's because we have the contamination of other kind of legume in the resident vegetation, and also nitrogen supply from cover crops changed during the walnut growing season, which we have more nitrogen available during relatively later season of the growing season, like in July, August. And the difference in organic nitrogen in the top 30 centimeter of the soil depths between the cover crops and the tree lines are quite different. They're in the opposite trend. And the leaching, nitrogen leaching potential, they might vary because with the tree age. So that really give us the some idea is about like when we doing the nitrogen management for orchard with different age, those kind of nitrogen demand or nitrogen management probably will be subject to a little bit of change. Okay, at the end, I would like to show you a very quick video about the overview of the project, and this is kind of required by the funding agency. Good morning, everyone. Welcome to the fair today. This soil health demonstration project focused on understanding how different cover crop mix impact the soil health. So in this project, we looked at like 3 different mix. First is clover, which have the different varieties of clover. And the second treatment we looked at the mix of clover and grass. And we have another treatment called multiplex, which consists of the vetch, fava bean, and mustard, and other kinds of legumes and species. So in this project, we investigated the greenhouse gas emissions from the different treatments. We also looked at soil biodiversity, and look at the soil nematodes and earthworms. We also look at the soil carbon sequestrations at the different soil depths. At the end, we try to conduct the economic analysis to demonstrate the cost and returns for different cover crop mix. We hope to develop the best management practices for cover crop management. The results of this. Okay. Great. Yes, that's concluded my presentation today, and I will be more than happy to take questions if we still have time. Thank you.
Thank you very much. Yeah, we're kind of tight on time, but maybe, of course, that's a very informative studies, and thank you for sharing that. Obviously, cover crops have some other effects. So the 2 questions we have are about nematodes, clover hosting and increasing nematode populations, and also about cover crops being sources of rodent pressure. So do you have any thoughts about those in terms of, did you measure anything?
Yes, so we did like that. As I mentioned in the video, this project, we also look at soil biodiversity, which include nematodes. We collect the soils each year after we implement, we terminate the cover crops, and to understand is like how the cover crops impact the nematodes populations and also earthworms. But I haven't got the results back yet. So I will share that data in the, maybe if we have the meeting next time, and we have more like an overview of the data on that.
Okay, thank you so much. I will move on from here in the interest of time. Thank you again very much, Xia, and